of the day. Of the conference, actually. So this is what stands between you and the award ceremony, and then between the award ceremony and the end, there's fabulous entertainment, food, and an open bar. So it is a great way to end the day. I'm so delighted to be able to, I'm sorry for anybody to know me, I'm Maura Grassi, I'm the Executive Director of the Blavatsky Fund, and for my sins, they make me run the biotech panels. So it's a busy few days for me. But I'm delighted to be able to introduce this panel, Path Forward to New Drugs, and our moderator, sure, and I'm going to get it wrong, Cheppy. I hope I got it right. I'm going to hand it over because you don't want to hear me, you want to hear these guys. Here we go.
Awesome. Thank you, Morag. And thanks for all of you for being here. I know it's at the end of the summit, and many of you could have skated out of here, so I appreciate you being here. So the title of the panel is The Path to New Drugs, which when I first read it, I was like, this is pretty amorphous title. But then I got comfortable with it because drug discovery and development is a pretty amorphous thing. All the points in the value chain that we have to go through to actually develop a drug are constantly changing. We're getting better. There are new and emerging technologies, and every time we solve a problem, by the nature of the DNA of the people in this room, then we reach for the next hardest problem. And so, sort of by definition, it's an evolving process. The one constant in drug discovery and development that I found is that it is never a linear process. I've never ever seen a plan for drug development that's actually ever gone to plan, and I attribute that usually to the fact that we're trying to modulate human biology. And if we're being honest with ourselves, our understanding of human biology is not really what we think it is, which means most of the time we're kind of flying blind. And in my personal opinion, the best way to navigate the inevitable twists and turns of drug discovery and development is to work with other people that you share values with. And that's why I love this panel and this Innovation Summit, because it's bringing together a community of people that share values. We're all in here in this room, presumably because we want to make human health better, whatever definition we have of that. And so I love the fact that we're bringing this community together with this summit. And if we have time at the end, I'll actually share sort of a personal version of that story that stems from Yale. So why don't we do some quick intros? I'm going to be very brief on my intro because people are not here to hear me talk. They're here to hear you talk. But you guys be more detailed. My connection to Yale is I did my PhD here 25 years ago in the chemistry department, 100 yards up the hill. I also took a couple of classes in the business school when I was here. That was my first exposure to business and finance, which to me at the time was foreign. I've then spent the next 25 years in the Boston-based biotech scene. I worked at a couple of larger biopharma companies, Amgen and Biogen, but I've spent the vast majority of my time in the early stage startup space. I've had the privilege and the opportunity to work on 8 startups at this point, 3 of which went public. 1 we sold to GSK, and the other 4, fortunately, are still private. So that's me. Jenna?
Thanks so much. So I'm Jenna Dorito, COO and co-founder of Revalia Bio, which is a Yale spinout, and I've been lucky enough to be in the Yale community since I was in undergrad over 10 years ago now. So Yale is, Revalia is a Yale spinout that is actually audacious enough to think that we can change the path forward to new drugs, and we do that by starting with different sources of human data. So my background has been in human organ perfusion for the last decade plus. I got started in departments of transplant in Cambridge in the UK, where I was learning how to bring organs back to life, primarily with the focus of bringing them back to life to preserve them more for transplant, to keep them alive longer, to running them on treadmills to see, can we make more organs available for the patients who are on the wait list? But in all of that work, we realized that those organs were going to get discarded anyway because they were from donors who were dying with diseases that we did not have cures for, and we fundamentally did not know how those diseases worked or how to cure them. So we really spun out of Yale. I was working as a postdoc here. We tried to make it work in an academic lab for many years, but we just got bigger than the small closet we were working in here to have a 24/7 operation that can say yes any time of day or night to these organs that were being thrown away anyway. So now at Revalia, we accept these organs, put them on pumps, bring them back to life. We get to work with pharma companies, biotech companies, med device companies who are looking to really have an insurance policy to know when they put their drug or their device in an organ, before they go into a living human patient, they can get human data and know that their drug is going to be safe and that it's going to be delivered the way they expect. So really excited to talk a little bit about the NAM space and the path forward to new drugs here today.
Thank you. Hi, everyone. My name is Katie McCauley. I'm responsible for portfolio strategy and discovery biology in a department at Novartis called the Exploratory Disease Area, or we call it DAX. So I am responsible for a portfolio of projects spanning a number of therapeutic areas and modalities. And really, I mean, in the spirit of thinking about this path forward to new drugs, my role is really to help teams think about where the science meets the strategy. So how do we do the right next experiment? How do we get the right set of data? How do we think about what models we need? What data we need to build confidence, to understand the risk, and to move new therapies forward for patients. So this is actually, I have no connection to Yale, but I'm really been thrilled to be here the past couple of days. I'm really interested in emerging therapeutic areas and emerging modalities, so it's been a pleasure to learn from you all, and I'm looking forward to learning more on this panel.
Hi, everyone. My name is Yu Chu. I'm working at AstraZeneca, currently as the Executive Director, heading an in silico sciences and automation team within the biologics engineering. So for my team's job is to develop or discover and develop biologics, antibodies, nanobodies for all different kind of therapeutics. My background is structural biology and computational biology. Lucky enough to work with AI scientists as well as the lab scientists to move forward with the in silico solutions from the basic lab automation to a database, and then to AI model training, and then to molecule design. So yeah, that's me.
So, hi everybody. I'm Derek Chalmers. I'm the co-founder, CEO of Oban Biopharma here in CT. We are very much focused on small molecule development. I know we may speak about other modalities here this afternoon. Surprise, surprise, we are focused on metabolic diseases as a primary therapeutic area, but also CNS diseases. Prior to that, I was a co-founder and CEO of Cara Therapeutics, also here in sunny Connecticut. And then prior to that, a co-founder of Arena Pharmaceuticals in San Diego, California. So the through line on all those companies, as I've always focused on a class of target, which is actually very familiar to Morag over there, who worked in one of the most famous GPCR labs in the world. So we spent a long time on GPCRs. We've put 4 drugs on the market. We put 21 into the clinic. And excited to be here and talk about it. Thank you.
Yeah, hi. My name is Mitch Taub. I work in drug development at Boehringer Ingelheim Pharmaceuticals, which isn't that far from here. I've been in the field in DMPK and also drug transport for maybe 28 years now. 25 at Boehringer Ingelheim and 3 before that at Novo Nordisk. I worked in Denmark. I'm also a traditionalist working on small molecules primarily, but I see a lot of the newer technologies come and go. However, being a traditionalist, I would say I'm surprised every time. We're focused in development, of course, when a drug comes from research and we enter our first human trials, getting it all the way to the finish line. And as much as I think I might know exactly what's going to happen for every new NCE, I am surprised every time. And so we're in the business of identifying new technologies to enable the work that we do to move faster, accelerated, of course, and get better results and mechanistic data that's going to support our filings and make better drugs.
Awesome. Thanks, guys. Just a little heads up that I'm sure a lot of you were at the 9 o'clock session this morning around sort of investors in biotech, and there are some overlapping topics that we're going to have today, so just a heads up on there. But it sort of makes sense because the issues that investors face in biotech investing are not different than the ones that operators face, and that's what this panel is, a group of operators. But I will tell you that in my experience, investors and operators see the world in a very different light. And so while I think a lot of the topics are going to be the same, probably some of the opinions that you're going to hear are going to be quite different. So the first one I'll start on, which is one of the elephants in the room, is artificial intelligence. It was talked about this morning. I was actually sort of surprised at the amount of skepticism on the investor panel about the value of AI, at least today. I was surprised by that. But I'm excited to hear from every one of the panel members today about their experiences here. So AI, there's been, it's now become almost a foundational tool in R&D, and there's been a ton of hype around its ability to shorten timelines, lower costs, and improve the overall efficiency of drug discovery in development. Everything from target identification to protein structure prediction, generative molecular design, toxin ADME prediction, even clinical trial design. And in fact, even the FDA is now adopting AI tools to help it read, summarize, and write regulatory documents. That's interesting, but also a little bit scary to me. But anyway, I would love to hear from each of the panel members, and we'll start here, your experience with AI so far in your roles, or not, and sort of where you see the promise or the hype in its ability to make our business much more efficient.
Yeah, absolutely. So I think, at Revalia, AI has been a core to the operations of what we're producing here. So we use it from everything and being a small startup to how we do HR compliance and making sure that our team members can ask questions and have, an agent be able to be there at any time of day or night that can query our handbook. But we also use it in how we're doing organ acceptances. So if the way that our company works is we are fielding organ offers 24/7. So we are evaluating them, what trials can they be enrolled to, and do they match certain criteria? And the way that worked when I was a PhD student is I was up for 3 years getting phone calls every hour, and I would have to go through a chart and make a decision within 30 seconds. And maybe I would miss something, or maybe I would just be too tired and not want to say yes to getting those organs in the middle of the night because there's a human in the loop that is exhausted. And so when we have that, when we don't have the safeguards around being able to have an objective yes every single time when the criteria match where we want to enroll, that is really reducing the human error and the human bias in our systems. So we have an agentic agent that is helping us screen all of our organ offers so our teams can sleep better at night. We have, agents who really help us put together and I'm talking about them as people because that's, how much they've really increased the value in our company. It's building these agents have also almost been like additional team members for us. But we're really transparent in the way that works with our customers. We're not going to say, here, you're buying this AI product. This is an AI-enabled product, and we want to be really transparent with that. We're not going to give anyone a black box solution where they don't know exactly how all the data was processed and the data came for it. But for us, AI really makes us more efficient as a company.
Yeah, I like that example, I think, because I think it really demonstrates how AI is a useful tool for synthesizing large amounts of data, for seeing patterns that maybe a human or a tired human can't see. And I think this is probably a lot of the promise that many of us see. I think for me, there's of course a lot of opportunity here, and I think it just is important to think about what data do we really have? I'm a molecular biologist. I think, to Stuart's point, there's many questions we still don't understand. And I think some of them we don't understand because we don't have the data, and some of them we don't understand because we're not seeing the pattern. And so I think learning how to work with these AI tools that are available and how we can use them to ask better questions of our data to get maybe different data depending on these evolving and new tools that are available, I think that's really where we'll see the progress be able to be made.
Yeah, for me, I want to characterize them or separate them into two categories. One is for infrastructure AI, which is sort of LLMs and AI agents that basically they can deliver you, connect you to the data, connect you to the tools to increase your efficiency, to increase the productivity. The other category, I will call it, is the specialized AI, which is going to be trained on specialized data, for example, structure prediction, for example, generative models for molecule design, for molecule optimization. That relies on a lot of domain data, which is not easy to get, right? So you need to generate those data specifically in certain conditions. So I think in the specialized category, we have some specialized within the company's data versus the public data, right? For MR sequences or DNA sequences. sequences, and general antibody sequences, general structures. There are a lot out there. So you can see the progress it's made so far is on the structure prediction, which is, relies on a lot of the PDB data, some of the language model based on the large sequences out there. So I think that's kind of falling to the public area. For internal data, I think we're still in the early stage. Basically, we're building the company infrastructure. So having an automated lab, getting the data automatically pushed into the database, I think is still under construction. So that's going to take quite some time to collect those data, set up the standard, and then getting the data in good shape for AI to learn it. I think the AI to learn from that infrastructure is relatively easy compared to building that database, building the lab automation, right? So I think that's going to take some time, but eventually, I believe that's going to give you the competitive advantage, giving you the power to make your molecule better, to make your molecule faster. So yeah, that's sort of my understanding. Yeah.
So I think I'm actually going to sound like an investor from this morning a little bit here. So thinking about this generally, and then I'll get to how we use it a little bit in discovery. So I think AI in drug discovery is a lot like the Scottish national football team. There's a huge amount of promise, a lot of talent working there, good strategies, but there ain't any more shots on goal than we used to have. Right? The real metric that counts here is how many successful therapeutics are we getting through the FDA. We're going to talk about the FDA a little later, but looking at NMEs for the last few years, I don't see a logarithmic shift in NMEs coming through. In fact, we had less last year than the previous 3 years. So undoubtedly, there's huge promise at the front end of this process. And there's no doubt the data looks like we can get to phase 1 molecules more rapidly, and that's where we use it. In terms of small molecule discovery, it's, in the good old days, chemical diversity was 500,000 compounds in your library. Today, we screen at OBAN about 20 billion in 48 hours, and that is unbelievably useful in finding chemotypes. And then the next layer on top of that, where AI is actually practically useful, is taking those chemotypes and running them through an AI-based model looking at ADME and PK. But that's all based on the database, and we spent a long time building that database, particularly for the class of target we're interested in. And I think that is valuable information. But that gets us to phase 1. If you look at the data, and there's some clever people from Boston that are pumping out reports every 6 months on this, the success rates at phase 2 are really not changing. The success rates at phase 1 are seeing a slight increase, maybe 50% in terms of efficiency. So we're getting there faster. And I think the molecules are, in general, a higher quality. But at this point, and I know that could change as this trickles through and the quality of the targets maybe gets better. But I think AI, for us, gives us faster, cheaper failures. And faster, cheaper failures are actually really useful in our business. We don't want to spend a lot of money on drugs that are not going to make it. But I think the promise is there, just like the Scotland national team. We might get to the quarterfinals one day, but we're not quite there yet. And I think it's going to take a little bit of time, and people building really specific databases for the class of targets that they work on to really get value out of AI. So I think it will come, but we're not quite there. Sorry about the cynicism.
Yeah, certainly there's a big push toward utilization of AI at Boehringer. I'm an early adopter when it comes to technology. I think about the first generation iPad years ago. I use AI. I probably use it for kind of boring stuff compared to what the other members of the panel have talked about, except the HR comment. So when it comes to writing performance reviews, it's incredibly helpful. When it comes to reviewing reports, incredibly helpful. When it comes to the member of my group who's just not all that great at English composition, incredibly helpful, because then I don't have to struggle with being an English professor and fixing the report before I can read it for content. So all of these things save time, and when I have more time, on that rudimentary stuff, then I can spend my time on the more important, mechanistic, scientific questions, trying to find answers to those problems. So hopefully we do see more success as we proceed through development. So yeah, it's a little boring. And I am an early adopter still, and I think there's a lot of promise, but I'm just being a little cautious at the moment, using it for what I think it's best at, and then I'm just going to wait and see how it can help me with the really complicated problems.
I am going to start watching Scottish football now as a proxy for the value of AI. All right, we're going to switch gears to, I'm going to lump together what I'm calling emerging technologies, but also new modalities. So when I graduated from here and went into industry, we basically had two choices. You got a small molecule and you got an antibody. So pick which one you want. These days, the world has obviously changed quite a bit. And we've gone from a place where we were sort of redirecting human biology to where we're now fundamentally reprogramming it. And we have all of these new tools in our tool chests, like ADCs, targeted protein degradation, which, by the way, was invented here at Yale in Craig Cruz's lab, T-cell engagers, cell therapies, RNA, gene editing, the list goes on and on. So I'm curious to know, and I'm going to start with you, Mitch. I know this isn't your job, but because of the nature of your job, you're relatively modality agnostic. I know you said you focused on small molecules, but where do you see, the most excitement in terms of new technologies or new modalities giving us new tools in the toolkit? And Katie, I'd also love your thoughts on that afterwards.
Yeah, I mean, every company is exploring all the modalities. In the last gene therapy discussion that we had in the last hour, I mean, there was certainly a lot of focus on that. Yeah, they're very exciting. I like learning about all of them. I do have the same questions that panel in the last hour came up with, though. Like, how are you really going to monetize this? Some of these can be very expensive therapies. In the pharma industry, we get slammed all the time for charging too much for our products, and I understand that. We want to make our products more accessible to our patients because we always have to be thinking about the patients in the end, and not just the people with the rare diseases and so on. But I think Boehringer and most other pharma companies do an excellent job at balancing it. Just like any other portfolio, you have to have your probably better quality shots on goal, to use the soccer or football analogy again, and then the long shots that might actually really pay out in the end and help people who have really no other hope. So I'm a fan of both. I work, of course, as I said, more with the standard small molecules, but I'll be keeping an eye on everything else as well.
Katie, thoughts based on your role in portfolio strategy, also relatively therapeutic area agnostic and technology agnostic. Any thoughts from you?
I think Mitch said it well, where I think, when we think about a portfolio of opportunities or ideas, I think having, more ways to go after the targets we're interested in, diseases where there's an enormous amount of unmet need. I mean, this in many ways is only a good thing, right? More opportunities. And of course, every modality and opportunity comes with its own challenges, its own caveats. And so we do think a lot about this from a portfolio perspective. How do we really bring forward the best possible medicine for patients, taking into account all of these factors, the right target, the right modality, the right patient population, the right biomarkers, all of these questions. So yeah, I think it's an incredible time to be working in this field, and exactly what you're saying, where it's fun seeing all these new ideas emerge, and I think we've learned about even more in the past couple of days that I hadn't necessarily thought about before. So yeah, I think it's a great opportunity to think about what else a medicine could look like.
Yeah, I can chime in. So I think I see the molecules as tools, and the modalities is going to be determined by the biological, mechanisms or even hypotheses. I think you see that antibody has been used as a targeting moiety that it can conjugate payload for ADC, for example, and then you see oligos being conjugated to it, peptide being conjugated to it. It's like delivering the payload to the right target. And the same for a cell therapy, AAV, etc. That's the best use of a binding moiety. But there is certain problems that biologics cannot do, right? Like a cell penetration, intracellular targeting, cell direct killing. I think their small molecule has its advantage. I think that's, but I think the future to me is like how you have a clear mechanisms of preventing disease or cure disease, and then build a best molecule, either like not even molecule, like cells or viruses that can do the right things for the right mechanism. That's going to be critical. And with the better molecule design tools, with more and more knowledge around different modalities, I think it becomes more possible.
Jenna, I got a specific question for you, because your role in your company is slightly orthogonal to what the rest of us are doing here with your work around human organs and transplantation. So I wanted to ask you a couple of things. One, specifically, I know you described it a bit, but maybe specifically a little bit more, how your work is actually helping with therapeutic development. But then also, I'll put you on the spot, I'm always interested in concepts like organoids and tissue on a chip technologies. So I'm going to put you on the spot on those and have you comment on those technologies as well.
Nice. So, I think with what we do is bringing these organs back to life to test very specific questions, right? And this is one part of what we like to call a larger human data stack. So when we're developing different therapies with these organs as a centerpiece, almost like a Rosetta Stone of this human data stack, we can get really clear answers in when I put my drug into an organ this way. Let's say I'm delivering a drug up a kidney's ureter. Where does it go? At what rate do I need to deliver it to? Because a lot of these drugs have been developed in animal models, and not surprisingly, pig kidneys are a lot different than human kidneys. Or sometimes we have another example. We've been working with a pharma company who's developing drugs that can be delivered into the tumor directly. And they were delivering them before they started testing them in human lung tumors. They were testing them because this is the only model that was available in either sheep tumors that weren't accessible or weren't reproducible to the human systems, or sometimes tomatoes, because we were like, maybe that density is the same density as a human tumor. We've found that human tumors have a variety of densities, and none of them are like tomatoes. So, we can ask really fundamental and really important questions of therapies. You can have the best therapy in the world, but if it's not going to get to the place that it needs to be, it's not going to be effective. And I think that's one of the foundational questions that we can ask in our whole human organ models here. But I don't think that is, this is not going to be a model, and Revalia is certainly not of the stance that this is the model that's going to cure all of drug development. This is going to be this single model that accelerates the timeline. That's not it. What we believe is we need human data, integrated human data, and a human data approach. So we think these organoid, organ-on-a-chip models are part of the solution. I actually think that all human data that is curated in clear and consistent ways with these new alternative models, the NAMs, they're all part of the solution. We can't develop a drug with a single page in a textbook, right? Organoids are a page. Organ on a chip is a page. Whole human organs are a page too. But when we start to use these human organs to seed the organoids, to seed the organ on a chip, to integrate with the patient data that's coming for it, then we have the whole book, right? We're not trying to create a drug based on the siloed information. So we think that all of these NAMs play an amazing role. Some of them have better scale than others. I can't perfuse 10,000 organs a day, even though they're out there to be perfused and people want to donate. But what we do really feel is these NAMs are going to have more consistent human data for us to develop. And with that, if we integrate it together, that's what's going to accelerate drug discovery and delivery in better, more efficient ways to make medicine safer.
Thank you. I'm going to shift gears toward lab automation. And I bring this up because I literally just had a friend who came back from China and told me that she visited 70 companies, 70, and she said all of these companies, they have people working during the day, and they have armies of robots working the rest of the day through the night, which is something you don't commonly see here. So I want to direct this toward you because of your roles at both AZ and Sanofi, part of which has been to implement new technologies to automate activities in the lab. And I'm curious to know your thoughts on how much of it can actually be automated that we don't need humans for, and what are the limits of that? Partially because I have 3 kids about to go to college and I need to tell them what to major in, so I need to know if they should be biologists or not.
That's really a dangerous question. I think what we have been focusing on is the biologics production and the analytics process, which is relatively standard. Because you have a general standard SOP already, so you know every single pipetting steps, how should you do it. And then that can be programmed through the robotic arms and high throughput robotics. That's the prerequisite that you can automate things. You have a standard SOP, you have a clear process, right? So for those, you need to have a lot of flexibilities, many things you need to change, especially different procedures. That's not going to be easy to automate. That's based on the current technology, especially with the like a humanoid type of automation. I think that opens a whole new door about how you can operate, even like building, I think, a physical automation with intelligence together. I think just one simple example for lab automation. There's no easy way you can open the door and putting a plate into an incubator, right? So you need to have people to do that. It's everything compatible with the robotic arm. You have to make it compatible. So in another sense that if you have a humanoid, then that can change the whole process dramatically, because you don't need to customize for automation specific solution. You can use that for adapting existing, workflows. That will give you the more flexibility. But regarding to fixed procedures, I think automation gives you the throughput. One side is you have a more data, more molecules to test with, and the second is the data is going to be more robust because it's eliminating the human errors and also standardizing the data format, which in the past has been a problem, especially for large companies with different size. With different scientists, they use different protocols, different buffers, different data format. I think with automation, there's a huge advantage to standardize everything. Yeah. I don't know if it answers your question.
Thank you. Yes. So I want to switch to therapeutic area of focus. And I bring this one up because our industry, one of the hallmarks of the biopharma industry is that we tend to go through these waves of therapeutic areas being in vogue and then very quickly out of vogue again. This happened when I was at Biogen. We thought we were going to cure Alzheimer's and everybody was into neurodegeneration. Turns out we didn't, and it was sort of uninvestable for a while. We had the immuno-oncology revolution with the checkpoints. For the next decade, everybody's chasing the next Keytruda. Didn't find it. Massive wave of failures. Seen that with cell therapy. Huge promise, then found out it's actually pretty hard to manufacture these things. So almost became uninvestable as well. And now these days you've got the GLP-1s and this revolution in metabolic disease and obesity, probably going to be the biggest therapeutic category ever. So I'm curious to know folks' thoughts, and maybe I'll start with you, Katie, because you're in your program management role. Where do you see the next wave of therapeutic area focus?
Easy question. Yes. No, I mean, reflecting on this a bit, I think anyone in this room would probably have their area that they think is going to be the next big thing that they're most excited about. And I think for me, what's important is not following, like a particular hype cycle. We're really excited about this idea, but I think what I see us really doing is thinking a lot about the mechanisms of disease. And I think we've been hearing about these opportunities with new human data. I think with AI, we see this ability to potentially really see patterns in data we haven't been able to see before. And so I think, we're continuing to learn more and more and more about what are the mechanistic drivers of human disease. And I think the next big opportunities are going to come from that deep understanding, from asking these questions in a way that maybe hasn't been possible before, from really pushing the edge of what we can understand in a lab, what we can understand in the clinic. And I actually think GLP-1s are a great example, a drug that obviously works incredibly for obesity, but also now being looked at and successful in other indications. Yes, obesity and metabolic related diseases like CKD and MASH, but also now people are thinking of GLP-1s for addiction, for other types of diseases. And so I think it's that question of following the biology, really understanding what is driving human disease, and I think investing in models and capabilities and biomarkers in ways of measuring this data so that we have those insights available to us. And then taking all of that and pairing it with people like the people in this room who really know how to move a drug forward. So not just ask scientific questions, but really ask the right questions and push forward a drug into the clinic, whether it's a low molecular weight or a new modality that hasn't been invented yet. But I'm sure I'm not the only one with an opinion on this subject. So
Can I add to that? I'm going to violently agree with someone for a change.
you say violently agree?
Yes.
Just checking.
So I think we're going to see a sustained interest in metabolic diseases for a while now, not just because of the size of the population, but as Kate's getting to there's an innovation here that we're just scratching the surface on in terms of applicability of that whole system. So, and this ties into modality. So we ended up with peptidic injectable GLP-1 drugs because the natural ligands are peptides, and us pharma guys like to emulate the natural ligands, the way it's gone. And those have been incredibly successful. But where that field is moving, and actually the reason I was late here today is trying to get a late breaker into ADA for next week, that whole field is going to move to multimodal therapeutics, partially because of the reason Kate's getting to. There's a biology here that we don't quite understand in the interplay. And it's going to come back to how we regulate our metabolic system. We don't secrete only GLP-1 to regulate our metabolism. There are 6, 7, 8 very important hormones. At this particular meeting, there are going to be a number of companies, including ours, that are going to represent data which actually activates multiple systems simultaneously and has multiple aspects of applicability. So it's not a question of weight loss with consequent benefit for comorbidity. It's actually attacking the comorbidity and the therapeutic. And that's such a huge area because those comorbidities, getting back to investors, are very costly in our society. And Novo likes to tout the number is going to be 2 billion patients by 2030, and that might be an underestimate in this area. So I think that field is going to dominate for a long time because the mechanisms work so well therapeutically, and the modality is going to change. I think it's going to switch to small molecule by various means. That's now possible. So the peptidic issue and what people have been talking about earlier in the complication and scalability and cost is going to go away in that area, and I think there's going to be a huge explosion in multimodal metabolic molecules, and it's going to start this year and get to the clinic in the next 2 or 3 years. So I think that's going to be the biggest area. So just wanted to get that.
Derek, I'm glad you chimed in on that one because I'm about to burden you with probably one of the more boring topics here, but around regulatory and policy in our industry. We're talking a lot about places where we've been able to innovate and make great progress. There's probably an argument to be made that the regulatory dynamic is one that we haven't really made much progress with. So I'm going to you, Derek, because you've navigated this process successfully in the past. Not all of us have. But can you point to some areas where we can improve, we can get better as a collaborator with Washington to make this process more efficient for all of us?
Yeah. I think Janet Woodcock is doing the rounds constantly talking about this and how she views molecules from the industry. And I think the take-home message from her is the FDA is never going to be as excited about your molecule as you are. And so the way to work with the FDA is full disclosure on any molecule. I mean, that's an organization that's understaffed. It's only got worse recently, and overworked. I think if we view them as collaborators, and we're not sending polished versions of our data sets to these guys who are mostly very, very good scientists and reviewers, and view them as collaborators, things move much, much faster at the FDA. And my favorite quote from the FDA, I think it was their head statistician, is with respect to sponsors, in God we trust, but everybody else has to bring validated data. and that's all they're looking for, is a validated data package that's explicit, and I think things go much more smoothly now. I guess the thing that's changed in the last 12 months out of that biotech White House gathering from, I think, a year and a half ago, is that the complaint we all had was, it's taking so long, the system's so slow, people are moving trials out of this country into China, and that's particularly sensitive for certain administration. And so the response from Dr. Makary was, well, maybe as opposed to the exception that we can get approval on a single phase 3, that could be the default. And I think that is a huge, huge gift to the industry, that we can run one registration trial. Having run a few of these programs, and the expense to run those 2 trials is enormous at the end of the day. And if we can get it reduced to 1, I think that's a huge gift for us. The flip side of that is it has to be a damn good phase 3 trial, with all the bells and whistles and interim analysis and so on. But I think that's going to make a difference in how quickly we can get a drug approved, and capital efficiency, which is always popular with investors at the end of the day. So there is a positive there, but at the end of the day, honesty is the best policy, I think, as Janet Woodcock often says.
Appreciate that. Yeah, Mitch?
Yeah, I would also agree. They're very good scientists at the FDA and other regulatory agencies for that matter as well. And one thing I would add to what was just said is that, sure, especially for NCEs, you can look at the guidelines, you can look at what they publish, and these are the things you have to do, and you can check the boxes and just put that in your filing and send it off and hope for the best. But what I've found is that if you do all that well, and then you add something else. So instead of saying, well, based on the standard tools and technologies that you list in your guideline, I couldn't make this work, I couldn't get you the answer that you wanted, explore something else. So in our last 2 filings, we actually used NAMs, experiments with very advanced models, to try to answer some of these questions. Take a little bit of a leap of faith, take a chance, generate some new data. And then what I have found, and I've talked with somebody that we hired at Boehringer who worked at the FDA for 10 years, she said, look, if you do all that right, and then you show that you're willing to go the extra step to make the drug safer and more understandable and better from a drug interaction profile, whatever it may be, then they're going to give you a little bit of a pass on some of the easy stuff. And I think that's fair. And so I would advise people in the room who are thinking along these lines to consider that as well.
Awesome. I do want to leave some time for questions from the audience, but I want to end on a topic that was also covered this morning in the investor session, which is around the China dynamic. And obviously their emerging leadership in drug discovery and development. There's been a seismic shift. I remember the days when China was considered a service provider with their contract research organizations, and that's where you would go for lower cost FTEs. Obviously, that dynamic has changed in a massive, massive way. To put some numbers on that, there was a stat article a couple of days ago that mentioned that in the first quarter of this year, between pharma and venture investors, we spent $60 billion licensing assets from China, and that is more than 10 times what it was for the entire year just 2 years prior to that. So these are big, big numbers. So I would like to hear from everybody on this one. So maybe, Mitch, you can hold on to that microphone and we'll start with you. But what's because this affects all of us, regardless of the company we work for or the role that we work in. This is a massively shifting dynamic. But how do you see this new role of China in drug discovery and development? Is this an issue that needs to be dealt with, and there are some that are calling for policy changes to try to stem this tide? Or is this an opportunity that we all really need to embrace?
I mean, I think we all need to embrace it, but in our own way. So in my area, what we're looking at China for, at least right now, is speed. They can do things very fast. Yes, they have robots that work all night. Yes, they have a lot of people. When we need the rudimentary stuff done very quickly, it's an easy place to turn and a relatively inexpensive place to turn. So there's no way you can ignore that. And then kind of like what I said earlier about the use of AI, that frees up my time to do other things that are maybe a little more complex, maybe require a little bit more finesse or experience. China will get there eventually, of course, but for now, at least from my perspective, it's the speed and low cost.
Yeah, I'd agree with all of that. In fact, all of the chemistry we have, medicinal, synthetic, is performed in China today, not just for cost reasons, but for quality and speed reasons. And I think it's superlative, absolutely superlative. There's one thing we didn't get to on drug discovery I meant to mention earlier, Stuart, in terms of advancements that are helping with small molecule development. I think structural data and analysis at a very high resolution, and we work with a very good group in Shanghai to generate this. I think cryo-EM data is absolutely superb these days. We get it down to a resolution that was unheard of even a few years ago, so we're essentially the width of 2 hydrogen atoms at this point. And that, with the right software, gives us an ability to generate small molecules of very high potency. So we can routinely get to picomolar potency with small molecules on our targets, which was really unheard of a number of years ago. So I think China is great. We use it every week, very high quality, very fast, and I think it's a tremendous resource.
I would like to comment on 3 aspects for China. One is, I agree with you, is the speed. The reason for the speed is, people work harder. There are a great amount of automation. I think it's also an ecosystem. With a lot of the CROs being local and then give you a quick turnaround time. That's basically moving things really fast. The second thing is, I think the talents. I think I saw the news. There are like 10 million the college students graduating every year, right? So there's probably 70% of them are like STEM students. So they're a great pool of talents, and they work really, really hard. So I think you can see from the AI, from the biotech, a lot of also people well trained from the Western world went back to China, lead the group from academic, from the industry. So that's why you see a big boom happened recently. And I think the third part is really I'm not expert in the space, but I heard there are some really flexibilities in like getting early proof concept data for clinical trials, like investigator-sponsored clinical trials that it can really be quite flexible and getting POC data quickly. And also the huge amount of patient population that's accelerating the clinical trial, patient recruitment. In addition, I think the patient's compliance is also way better than other places. So overall, I think this is a great opportunity for the biotech industry, for the pharma industry, to embrace the capabilities, the talents, and also infrastructure in China to moving, because this is eventually drug for all the human.
I don't know if I have anything to add after that. I think I would just echo what others said. I think it's a great opportunity to learn, to collaborate. I think there's so many amazing things happening in China that we're seeing. So again, honestly, you just said it super well, so not a lot to add, but I don't know if you want to say anything.
Yeah, I think there are amazing use cases to lean on China for, and we also have to make sure that there's infrastructure that can protect against when there are tariffs or when there are supply chain, interruptions. But the one thing that I will say, being a person who really cares about human data and the integrity of that, is that I think generating that very unique human data that comes from samples from patients is something that's uniquely American. We have that, of course, throughout all the world. We have biobanks, but what we're able to do in the U.S. that's unique is use those samples for commercial entities. That isn't true throughout Europe, and while it is true in parts of Asia, there's always been some kind of questions of ethics behind where those samples are sourced from. And so what we are doing in the US is really creating an infrastructure of integrity, of making sure the patients are informed, because it hasn't always been that way. And when we move to a path of developing new drugs that's based on human data, we need to take the donors along with them. We need to make sure that they have legacy, that there's integrity in each of those samples, that there's tracking. And parts of Europe have been doing that really, really well. But I really think that with the scale that we have in the US and the infrastructure that we're setting up here to enable that, that's a uniquely American solution that can help accelerate drugs on this new path.
All right, maybe I'll bring it back home with my personal anecdote of the value of the Yale community, and then we'll open it up for questions. Almost exactly 10 years ago, I was working at a venture capital firm in a role called an entrepreneur in residence, which doesn't mean anything other than help them start a company and then run it. And they gave me a task, which was to start a company. And I said, that's it? Like, that's the amount of guidance that you're giving me? And they said, yeah, that's it. Go figure it out. And the problem was I didn't have any good ideas myself. And so I sort of struggled for a while to try to find a good idea. Eventually I got smart and decided to get on the train and come down to New Haven, where a good buddy of mine who I went to graduate school with here stayed on as a professor in the chemistry department. He's a lot smarter than I am. We sat down and we had lunch one day, and we hatched an idea for a company that is now called Chimera Therapeutics. It was one of the earlier protein degradation companies. Fast forward to today, it's worth $6.6 billion and has late-stage clinical trials for what's going to be very likely new medicines for hundreds of thousands of patients suffering from autoimmune disease. That came from a conversation in the Yale community. I had lunch with him again today. We talked about a bunch of new ideas, so maybe there'll be the next one coming out of it, but appreciate events like this, and there is true value to doing this. So thank you all for coming. And if you have any questions, please just raise your hand.
Thank you very much for sharing all of this really wonderful information with us today. A couple of hours ago, some of us were in a session related to how important it is to incorporate patient advocacy organizations or patient advocacy work into R&D activities. And so I just wondered if you could share how you think about incorporating patient perspectives and patient needs in the early R&D phase.
Well, I can say something about that, but the last drug we put through the FDA was specifically for dialysis patients, and we brought them on board very early. A couple of advocacy groups in that community, and that was extremely helpful moving forward, and all the way through the process with the FDA, and the commentary from those groups and supportive of the drug and the need for the drug. Really scored points with the FDA. So we brought them in once we had the first meaningful phase 2 data set. We were in communication with patient advocacy groups at that point.
Maybe building on that a little bit, I think this is such an important point, and this is something that I think we're seeing, again, more and more of, is really hearing the patient voice in early drug discovery, drug development. So I think we're really, I think many years ago I felt like I would hear a lot of assumptions about what patients wanted or what, this particular patient population would need. And now I feel I'm a part of a lot more conversations where we're really talking to patients, working with advocacy groups, really trying to understand, like, what do patients need? Because no one knows their own health like themselves, right? Like, people know what they want, and yes, talking to a single person, it's not statistically significant. You're not going to, run it through AI and get some insight from it, but you're really going to learn about what that person is feeling, what they're suffering from, what it's like with their family. You're going to learn about how the burden of having 2 young children and having to go to the hospital every week for a treatment. You're going to learn these stories, and I think this is where we then see where are the opportunities, what drug would make a difference for these people. and so not only does it help inform our science and I think our decisions, but also just, I think for many of us, it's what gets us up in the morning and going to work. I mean, I think it's incredible. So.
Yeah, at Beringer, we have quite a patient awareness program. And that wasn't something that was there when I first started. This is something relatively new. And I've had the opportunity to meet some of these patients, meet the close relatives of some of them. One guy's wife even came up and gave me a giant hug at one of these lunches where we got to meet them. I didn't even work on the drug, but she was just, crying and thanking me, and I was moved anyway. So it's, I think, really important when you can be kind of anonymous working in a small or large company to really experience the stories of these people who are affected, and certainly the advocacy groups, the people who are looking for new medications. That's super, super important. So I think we should all be aware of it, and hopefully all of our companies promote that.
Maybe something just real quick is, so the way we integrate that into our workflows are interviewing sometimes the donors firsthand, but definitely the donor families. And that's actually how we decide which new areas to go into. So when there are more donations in a field like lung cancer and it disproportionately affects a certain population, like we have a program out in Oklahoma where there's still a large community of heavy smokers. So there's a higher incidence of lung cancer that affects the whole community. And in Oklahoma, there is not almost a single person who doesn't know someone in their family or a friend who's been affected by lung cancer. And that is really what pushes us to start new programs within Revalia, where there's donor advocacy, where there's donor family advocacy, patient advocacy that we're involved with, and there are people who want to donate but can't. That's what really fuels all of our work.
Thank you all. I've been told we have to wrap it up because we've got some awards to give out. So thank you all for being here. And if there are further questions, feel free to just come up and approach us.